Hello, everyone. Thank you for attending today's webinar on the comparative study of animal-derived ECMs and VitroGel Hydrogel for hiPSC maintenance and intestinal organoid generation. My name is Christopher Oak with the Well Bioscience, and I will be your moderator along with co-moderators Dr. John Wong with the Well Bioscience and Dr. Rodrigo Cristofoletti from the University of Florida, along with our speakers, Dr. Janie Jans from the University of Florida. During the webinar, you can ask questions using the Q&A tab. We will collect them and present the questions to our speakers at the end of the session. Also, we have polls during the webinar, so feel free to participate. So let us get started. I would like to introduce our speaker, Dr. Janie Pinheiro Jans from the Department of Pharmaceutics at the University of Florida. She joined Dr. Cristofoletti's lab at the Center of Pharmacometrics and Systems Pharmacology to develop and optimize organotypic and microphysiological systems for the in vitro testing of pharmacological compounds. Dr. Jans, I will turn it over to you.
Thank you so much, Chris, for that nice introduction. Hello, everybody. As Chris mentioned, our laboratory is part of the Center for Pharmacometrics and System Pharmacology at the University of Florida. And in our lab, we focus on incorporating human-relevant in vitro systems with computational models to improve early stages of drug discovery. Today, I will be talking about a study that we performed in which we compare using animal-derived basement membranes with VitroGel hydrogel system to maintain human-induced pluripotent stem cells and to also generate intestinal organoids. So to start with, I want to highlight how tissues throughout the body have different composition, architecture, and mechanical properties. And even within the same tissue, there is some heterogeneity. For example, comparing tissues on the brain with other type of tissues like a skeletal muscle or intestinal tissues. Brain tissue has a large amount of non-fibrous components like proteoglycans, and these ones lead to stiffness values that are in the range of 1 to 4 kPa compared to muscles or intestinal tissues where have more collagen one and have a higher stiffness values. And when we talk about the stiffness of a tissue, it's good to note that it's very important to pay attention to the methodology and the mathematical equations that they use to actually come up with this number, because you're going to find through literature different values for specific tissues, and it's good to look at the range or the range of values that is reported for that. But not surprisingly, the different properties of the tissues can guide cellular behavior and fate. Cells are mechanosensitive, and this means that they can actively sense their surroundings by exerting contractile forces as shown here in D. And this mechanical stimuli is transduced or converted into biochemical signals such as activation of signaling pathways. Cells also have a social life. So they interact with the different components that surround them, or the extracellular matrix, and they interact with each other. And these interactions are through adhesive junctions, as shown here in C. And most of these adhesive junctions, a large part of them, are part of the superfamily members of integrin. So these integrin-mediated connections between the ECM and the cytoplasm of the cells regulates key functions such as migration, proliferation, and also differentiation. So the composition of the extracellular matrix as depicted in A, the spacing between the adhesive ligands as depicted in B, and the stiffness of the ECM as shown in E, all these regulate cellular fate. And these are key aspects to take into account when we're selecting a biomaterial system for a stem cell culture and organoid generation. So for today's webinar, I want to first start by highlighting the importance of the cellular microenvironment when guiding cell fate. And then I will share with you some of our work comparing the workflow using a classical animal-derived basal membrane systems with the Hydrogel, VitroGel hydrogel system for both human induced pluripotent stem cell work and generation of organoids from these ones. And I will wrap up with some aspects to consider when deciding the type of system to select for your work. So, as I mentioned before, the composition of the ECM varies with tissue type, but it also changes with disease stages. And this has an influence on the cellular function. Here is an example of a recently published paper. It was published last year in Cancer Cell. And this team looked into the collagen 1 that was made by pancreatic cancer cells and compared with the collagen 1 that was made by the activated fibroblasts that normally make this stromal tissue that surrounds pancreatic tumors. So what they found was that in contrast with the normal collagen 1, that is a thyroid primer composed of alpha 1, alpha 2, alpha 1 units, the one produced by pancreatic cancer cells, it's unique because it's a normal trimer only formed by alpha 1 units. And this one promotes oncogenic signaling, leading to cancer cell proliferation. So when people from this team went ahead and deleted this collagen 1 homotrimer, the overall survival of the mouse animal model increased. So two key things to highlight from this is that the ECM is different in healthy and diseased tissue. And also that not only the composition of the extracellular matrix matters, but the quality of each individual component is very important and can influence cell signaling and cell fate. Another aspect to take into account is the stiffness of the substrate. Cells are mechanosensitive, and they sense the stiffness of the place that we use to culture them. They sense the substrate that they have on their floor of about 10 to 20 micrometer deep. And these mechanical signals can guide the differentiation of stem cells, as we can see from this hallmark paper, where Engels showed that human mesenchymal stem cells could be differentiated into 3 lineages, neurogenic, myogenic, and osteogenic, only using the stiffness of the substrate. So changes in the matrix elasticity can affect biological functions of cells and tissues, and we need to consider this when working with the stem cells and organoids. So basement membranes extract from murine sarcoma. It's the classical system used for iPSC work and organoid culture. However, there is many other systems that have been used through the years. The classical one, as I mentioned, is Matrigel, is an example of the basement membrane extract. But other systems that have been used are, for example, systems based on collagen hydrogels, which have tunable stiffnesses, or decellularized tissue that provides organ-specific biochemical profiles, synthetic matrices that have a well-defined composition and tunable mechanical properties, and of course, there is other systems that have been specifically optimized for very specific applications like bioprinting. So in our study for this specific case, we used 2 commercially available animal-derived basement membranes, the classical Matrigel, and we use also Cultrex. These 2 are based on the same system. They are extracted from mouse sarcoma. And we compare using these 2 systems for stem cell work and organoid generation, specifically intestinal organoid generation, with using a VitroGel hydrogel system. So the idea is we're trying to decide which one to pick, and how do we decide which one to pick? So we started with the classical protocol to coat the tissue culture plates with animal-derived basement membranes, which involve diluting the highly concentrated aliquots according to the lot specification sheets of each product. And of course, we make sure to work on ice to prevent any early gelling. And we made the solution, added the solution to the plate, and after waiting 1 to 2 hours, we added then the cells with the media to the plates and culturing them in there. So we use 2 human-induced pluripotent stem cell lines. Different lines of cells, we have a slight variability in terms of the growth rate and differentiation, and that's fine, that's normal, but we want to make sure that the workflow that we use is reproducible for different cell lines. So that's the rationale behind testing this with 2 different cell lines. Both are commercially available. And we went ahead and checked the expression of a stage-specific embryonic antigen 4 and found that there was no significant difference between the cells cultured on Cultrex or Matrigel, suggesting that these 2 basement membrane systems are equivalent. So in parallel, we went ahead and used VitroGel Hydrogel matrix to culture these same iPSC lines. On top of the Hydrogel, there is different methods that we can use this product. In this case, we decided to use to culture the cells on top 2D method to make it more comparable with the protocol that we use for standard animal-derived basement membranes. So we first mix the, we're using MTSER plus as the media. So we first mix the MTSER plus medium with the hydrogel solution and then added to the well and waited 10 to 15 minutes and added the cell suspension on top of that. All this work is done at room temperature. We don't need ice for this. So the first thing that we noticed is that the morphology of the iPSCs colonies in VitroGel is different than the ones on the other basement membrane systems. And this is okay. Some people might freak out first, but it's fine. These cells are sensing the soft hydrogel underneath. And because of this, they form these round colonies with less spreading. And also because this is a hydrogel system, it would tend to swell a little. So the colonies that are now more of a 3D colonies are going to be semi-embedded into the gels and take this morphology. However, we also noticed that there were some areas that have what indicates of more cell death. So we went ahead and went back to actually making the hydrogel. In this case, we make the hydrogel with a higher concentration of growth factors. So MTSER Plus comes as the base and medium and the supplementation. So what we did for this new hydrogel, we went ahead and made the medium that had 3 times the concentration of the supplemented part of the MTSER Plus. And we followed the exact same thing. We just have the medium that we used to make only the medium that we used to make the VitroGel. Hydrogel with 3 times the concentration of growth factors. And what we found was that there were more colonies growing, less cell death around those colonies, and also we see a higher expression of a stage-specific embryonic antigen 4, and with values that were on the same range of the values that we got for the animal-derived basement membranes. So to us, that was great news, but it also suggests that we could have a full control of the growth factors that we add into this hydrogel system. In this case, we're using MTSER Plus. It comes as a kit, but the same logic applies for in-house made medias and cocktail of growth factors. So you can just increase the concentration of the growth factors. And there's multiple options. I heard that as well. The World Bioscience will be launching soon a new supplement that can be used for cell suspension and also for cell culture on 2D and 3D. So going back to the key aspects to consider when selecting a biomaterial system for a stem cell culture and organoid generation. Two-dimensional cell cultures has been and is still extremely helpful platform, but it does not truly reflect the physiological three-dimension environment of the cells. In 2 dimensions, the cell matrix interactions are restricted to the planet directions, while in 3D, cell adhesions are distributed in both planar and perpendicular directions. Many of the research have also reported that 3D cells form less stress fibers, so they are going to be less spread. And the secreted factors can be highly concentrated compared to the 2-dimensional conditions. So 3D culture for stem cells provide a physiologically relevant microenvironment. And following this idea, we can use products like VitroGel, Hydrogel, and customs of this product like VitroGel, Hydrogel, for stem cells, which has been specifically optimized to sustain stem cell culture in 3D. And we can mix up with other technologies. In our case, we specifically look into combining these hydrogel systems with micro devices that have a controlled geometry to investigate the effect of the geometry and the stiffness on crypt villus formation during intestinal organoids generation. This is an ongoing project in our lab, and we would love to share more about it in the future. I just wanted to mention here another application for these products in 3D cell culture. So going back to the working with iPSCs, our ultimate goal is not only work with the stem cells, but actually use these induced pluripotent stem cells to generate organoids. And that's exactly what we did. We use a commercially available kit from Stem Cell Technology. This kit was developed based on a paper published back in 2011 by Spence and coworkers. And the workflow to differentiate iPSCs from stem cells into intestinal organoids is based on 3 main stages, with the first being differentiating the iPSCs into a definite endoderm, and then moving on to the mid-hand gut. And from this stage, as first, they're going to start detaching from the cell culture, and we can collect these spheroids and embed them into an ECM system, classically the DOM system of, and then mature these spheroids into intestinal organoids. So that's exactly what we did. And we want to highlight from these 2 main points. So the first one is that regardless of the matrix system that you use, an efficient generation of intestinal organoids, and I think of organoids in general, requires studying with a culture of iPSCs that has minimal spontaneous differentiation. So because of this, we compared the expression of a stage embryonic antigen 4 on these iPSC culture on the different systems, and we found no significant difference between Matrigel, Cultrex, and VitroGel matrix, the one that we generated using 3 times the concentration of growth factors. So the second point that we want to highlight here, it's going back to the difference in morphology. These semi-embedded clumps of iPSCs that are in VitroGel will grow through the different stages of differentiation as a semi-3D structure and lead to the formation in our experience, of larger organoids or spheroids. I'm sorry. And these, instead of like spheroids that detach from the culture and embed on the ECM, we were seeing larger spheroids forming, and we did the exact same thing. We collect them and embed them into the ECM system. And so for this step of embedding the spheroids, we're using a VitroGel organoid system. So this is VitroGel. We use a VitroGel discovery kit, which comes with 4 different formulations of VitroGel. And these formulations have slightly different properties, mechanical properties. And what we did was following the same idea of making the VitroGel hydrogel. In this case, with the medium that we are using is a stem-diff intestinal organoid grow medium, and we generating this medium using increasing the concentration of the supplements or the growth factors. And we made the hydrogel. We mix the hydrogel with the spheroids in this case, and we seed them. And we use the done method because it's the more classical protocol that has been used for the other basement membrane systems. So we wanted to keep this consistent, but there are several ways that we can use this product. So we did that, then we added the media. All the workflow is at room temperature, differently than Cultrex and VitroGel, in which we make the done, but we have to work on ice. And so this is representative images of growing the spheroids and generating these organoids in the different VitroGel organoid systems. And what we did was we look into the growth of these organoids. So while we start the culture with a relatively similar size of organoids, after 5 days in culture, we're seeing some variations in the average size of the organoids, with VitroGel organoid 3 leading to the largest intestinal organoids. And we speculate that the different mechanical properties of the VitroGel organoids hydrogels might play a factor in here, with VitroGel 3 being the closest to the range of a steady-state stiffness that have been reported for unaffected human intestinal tissue, which is around 800 to 1300 pascals. And to put this in the context of cell culture material, standard polystyrene plates are in the order of 10 to the 9 pascals. So we also compare using VitroGel organoids with the classical Cultrex and Matrigel. And again, we start with relatively similar sizing of organoids, and after 5 days of culture, they grow at different rates. But we want to highlight that compared to the VitroGel 3, which is the one that we found to generate larger intestinal organoids from the VitroGel products, if we compare this one with Matrigel and Cultrex, we don't find a significant difference in the size of the organoids after 5 days, suggesting that these 3 products are might be equivalent in terms of culturing intestinal organoids. And I highlight that because for other system of organoids, you might want to optimize and find the specific product that works best for that one, depending on the properties. So putting all together, both system, animal-derived matrices and xeno-free hydrogel have advantages and disadvantages. Ultimately, the choice depends on the research question, and you might want to consider the end goal of your research and the regulatory requirements that might come with that goal. For example, in cases of comparing in vitro experimental results with a mouse animal model studying tumor microenvironment and cancer biology, using a matrix that is derived from a mouse sarcoma can be a great starting point. However, if your research question is more on the area of clinical application in humans, drug testing in vitro, you might want to consider xeno-free systems like VitroGel. And yeah, all systems have pros and cons. You need to consider these ones. For some people, they mention a lot that the lack of growth factors might require more supplementation. However, from our perspective, we're seeing this as an advantage because we're seeing it from the perspective of starting with a clean system in which we can control the growth factors that we add in there. And we could potentially investigate more mechanistic questions related with that by knowing exactly what is in our system. So ultimately, it depends on the research question. So our recommendation is to start with the end in mind. And I'm going to put an example. As I mentioned before, in our laboratory, we focus on incorporating in vitro systems with in silico models, such as physiological-based pharmacokinetic models, to improve the prediction of drug absorption, toxicity in humans. So the motivation of our research is the high attrition rates in drug development, which are mainly due to limited translation of efficacy and safety data from animal studies to humans. So, for example, here is a study that compiled the attrition data for oral small molecule drug candidates for development during the times of the years 2000 to 2010 in 4 large pharmaceutical companies. And what we're seeing from this data is that clinical safety and insufficient efficacy are the main reasons for failure during phase 1 and phase 2, respectively. And this highlights that the limitations of translating preclinical animal data to humans. So because of this, we specifically use microphysiological systems and organotypic models for a variety of projects in our lab. So a system like VitroGel that provides control of physical properties, composition, and also we don't need to worry about xenogenic effect of the animal product on the cells, provide us with a platform that aligns one with the efforts of reducing animal testing, and also it helps us to facilitate providing specific information when dealing with potential regulatory agencies in terms of like what is our system, because we know exactly what is in there. It's defined from the beginning. And so the overall take from this is start with the end goal in mind and optimize the system that you use for your stem cell work and organoid work early on, such that it allows you to achieve that goal at the end. So I want to thank you all for coming here today for the time. And also I would like to mention that there is a Jovi manuscript and video on the cooking that will show more details about the workflow that we're presenting here today. And with this, I want to pass the floor to John. I think he's going to tell you a little bit more about some of the VitroGel products.
Thank you so much, Dr. Yang, for this fantastic presentation. I just want to take a minute to recap all the VitroGel product has been mentioned in this presentation. So when we have these synthetic hydrogels, it's not like easy to make. A hydrogel has many different compounds inside. We try to build it from the bottom up to make it pure. But also it's adding all these different functions, different mechanical properties with this hydrogel. So we have the hydrogel systems with the different property for different purpose. And as mentioned in these presentations, when we use for these 2D hydrogel coatings, this VitroGel hydrogel matrix has been mentioned that using it for the 2D coating and adding the cells on top to maintain the iPS cells. We also have another product called VitroGel Stem, which we optimize the formulation to support the 3-dimensional stem cells expansion. And these products can support the stem cells directly from liquid nitrogen. So you can take the cells directly from liquid nitrogen and get rid of these cryopreservation medium and directly putting in this hydrogel system with the regular cell culture medium. The VitroGel organoid kits, again, for a different type of organoids, the mechanical strength and the hydrogel property needs to be fine-tuned for different purpose. So we put the 4 different formulations in this starter discovery kit for the customer to screen it. And so this is the hydrogel system that we designed to support the different type of the organoid growth. And I want to also mention that one of the complementary products, what we call is the VitroGel organoid recovery solution. So when people try to harvest the organoid out of the hydrogel systems, we do have these recovery solution which can work on both animal-based ECM or our VitroGel system. And the benefit of this medium is that it takes very short periods of time to dissolve the hydrogel, either for the like animal-based ECM at low temperatures, take around like 5 minutes to dissolve everything and harvest the cell out, which is very fast compared to other standard protocol. But it also takes very fast, around like 2 minutes to dissolve the VitroGel, and you can get the intact organoid out of the hydrogel matrix. And if you need to like further dissociate the cells into like small cells or single cells, you can add the cell dissociate solution. And as mentioned in the presentations, there's upcoming products for the supplement system that can help the scientists to grow these stem cells in our hydrogel system. So the supplement going to fit for both 2D coating culture, 3D culture, and 3D suspension culture. So that is another system that is coming up. If you need more information, please visit our website, and you can also scan the QR codes on this slide to follow up on LinkedIn for more products announcement. And also we have an upcoming webinar series about more learning series by using the VitroGels for different applications. So with that, I want to thank you for joining this webinar, and thank you for Dr. Yang for this wonderful presentation. And we would like to take any questions.
That's right. We're going to go to a Q&A session here. I think if we're going to go in order here to Janie here. One of the questions are, just for clarification, did you increase the growth factors in the culture medium or mix them with the VitroGel?
That's a great question, actually. So you can do both, but what we did here was just increasing the growth factor concentration on the VitroGel. When you make the hydrogel solution, just you have to mix the hydrogel solution with your media. So that media that you use, just have it that has higher concentration of the growth factors. You don't need to add extra growth factors to the media that you feed the cells later on, just the one that you do make the hydrogel with.
I want to add one more point on this one. It's because that the standard mixing ratio of our hydrogel is 2:1, which means that we use 2 parts of the hydrogel and 1 part of the cell suspension. And so as you can see that when we mix this, if you use the regular cell culture medium, it actually be diluted to 1/3 of all the growth factors in this cell suspension medium. And that is why we emphasize in our standard protocols that you need to increase the growth factor in this cell suspension to 3 times. So when you mix a 2:1 ratio, you bring it back to 1x.
Yeah, that's great, John, because like it's really easy to just like use the empty cell media or whatever media you're using, and you have to think that you're gonna actually be diluting it with the VitroGel. So if you want the same results that you see using normally the media, that you need to take that into account and add a little bit of growth factors to account for that dilution. Yeah.
I think this might ties in with the same question, but John, maybe for you, which other media can we use with VitroGel?
Yeah, and for these particular stem cell cultures, we can use like almost any kinds of the commercial medium or like the medium you make in house. The hydrogel system compatible with like MTs, the EA medium, and or other like stem cell culture medium. And the same concept can use for like regular mediums. If you don't use it for stem cell, you use it for like a cancer cell types or other cell types, or even like a tissue fragment. You can use the hydrogel with DMEM, MEM, DMEM F12, RBMI. Those are very typical medium system.
Yeah. For Danny, one question was, they don't work with intestinal organoids, hence the questions is, are the sizes of the organoids integral for the study of their function, or are you trying to gain a more uniform size of organoids?
Okay, so I would say the short answer is both. A little bit longer answer, both are related. So let's actually, I'm gonna go back to this slide and to answer that. Okay, there we go. Okay, so I'm gonna go back to this slide. So we're measuring the size of the organoids as one of the metrics that we can use to see how are they growing in these systems. But we are also trying to tackle one of the issues and classical issues with working with organoids is that normally there is a size distribution within them. And if we want to use this technology, let's say for more applications on the pharmaceutical industry, we need to make sure that we have a technology that is up to the standards of the pharmaceutical industry, and that we can reproduce multiple times these organoids. We can make them in the same way, and controlling the size of the organoids is one of the important things, and that's actually one of the motivations between these. For the research that I mentioned here, it's kind of like being able to control the geometry and control the size of these organoids is going to also be important later on when we're going to use these systems for testing different drugs. So I hope that answered that question.
Yep, I think it's good. And I think since you're on the slide here, Danny, one more question is, are the data for the intestinal organoids, the last produced slides of the data, are those colon or small intestinal derived?
Okay, so that's another great question, and I'm gonna go back to this slide here. So I mentioned that the protocol goes through 3 stages. So the last stage, the mid-hind gut portion, this is important stage in terms of the type of intestinal organoids that you're going to generate from here. So the time that the cells, regardless of the system that you're using, the time that they are exposed to the mid-hind gut medium is going to define the specific type of organoids that you're going to be getting, if you're going to be getting more colon organoids or more small intestine organoids. So that's going to be defined by the time that it's exposed to that. So for this study, what we did was we were collecting these fluids at a relatively same day, so we can compare we can compare one system to the other. So we wouldn't be characterizing, let's say, more organoids that are representative of the colon section with organoids representative of another section. So we tried to keep that date constant. So for these organoids, most of the spheroids were collected between vertical wells day 5 and 6.
Okay. And let's see any more. And we have so many more, but here we go here. What does recluption? Okay. And this is for John, I believe. A few things here for what is the chemical nature of hydrogel, and what does recovery solution dissociate ECM and also organoids for a single cell solution?
So yeah, this the hydrogel system is the synthetic polysaccharide-based hydrogel, and when we put the hydrogel systems together, we also modify this hydrogel with the different mechanical strength, function like, and degradability, all these different factors. So we try to make it to mimic the real microenvironments of the human microenvironments for the different tissue to growth. And in terms of that, the organoid recovery solution, the solutions will dissociate the hydrogel, but it won't break the organoid into single cells. So the idea is that we want to use a solution that we can take the intact organoid out of the hydrogel matrix. And if the user need to dissociate the organoids into a small fragments or single cells, they can simply just use some cell dissociation solution to do that after they take it out from the hydrogel.
Another question is VitroGel hydrogel already applicable for 2D iPSC derived cell cultures, for example, for imaging of plain cells? or more for study 3D culture systems? That's
That you means the VitroGel stem?
I'm not sure which one, but is VitroGel Hydrogel already applicable for 2D iPSC derived cell cultures?
Yeah, so we do have multiple cell culture methods. So the one method that mentions in this presentation is that we using the hydrogel, we mix it with the cell's culture medium with the higher concentration of the growth factor, and we put it as the one layer. And depends on the thickness of these layers you're coating, you put the cell directly on top, and then you can close that. And that is very standard methods for us, what we call is the 2D hydrogel coating methods. And we also have our standard methods to embed in the hydrogel for 3D cell culture, not just for the stem cell, but also for other cell types. We can embed in the cells inside of the hydrogel for the typical 3D cell culture. And for the 3D stem cells expansion or like a scale-up cultures, we also use the methods what we call is the static suspension culture methods, which we use the hydrogel to mix with the stem cells, and we further disperse this hydrogel into a cell culture medium, so the whole cell can suspension in this hydrogel system and keep growing in this way. So the idea is that we can use these 3D cell culture systems not just using the well plate. You can use the well plate or like a typical flask to culture it, but you can also use the same way to directly scale up to bioreactors to scale up these stem cells and culture the stem cell directly in the 3 dimension.
Thank you for that. Another thing I think was for, I think it's for Janie, won't this change in cell morphology when cultured in VitroGel have implications for the functionality of the cells? She said that I know the SSEA4 was good, but what about other things? Morphology is not important in the final application.
That's actually a great point. Morphology, it's one of the metrics. It's good to always, if we see a change in morphology, it indicates that there is some other functional things that are changing, but the key aspect is actually checking markers. So we check the C4 as one of the markers there, and we actually check others in addition to that one. For this presentation, we're just showing that one, but we highly recommend to check others. And sure, morphology is not everything. It just indicates that there's actually functional things changing into a stem cells behavior, and it will definitely change the behavior of cells. Now, what we need to think about is like, are these cells in just the native tissue, are they in 2D or in 3D? So if we want to look at a more biomimetic systems, those changes might actually be more representative of what they are in native tissue.
Another question that could be both for you. Have you done a proliferation assay or cell viability assay, I guess human intestinal organoids grown in VitroGel, in comparison to Matrigel and Cultrex?
So for viability assays, and I think you mentioned proliferation, variability. Well, viability assays in general for the stem cells and for organoids, you can do them. You can check for them, and yes, of course, we need to make sure that we have organoids that are alive and the cells are fine after. So yeah, for us, it works fine. Now, John might be able to tell more about specific, like working with VitroGel, because I mean that's their specialty. But so far, viability assays, there's a variety out there, and they are like with these systems, with those like animal-based systems and like VitroGel systems, they are friendly to these assays. So.
We, in terms of the viability assays, we do have another product called Cytos 3D Live Data Assay, and that is another product we optimized for the 3D cell viability testing. The product is actually very simple. So you just need to add like 2 microliters of these solutions into a 100 microliter system, which includes the carbon medium and hydrogel. And so it can penetrate the insides and so get the imagings of the cells of showing like live in greens and dead in the red color. So that is a simple and powerful tool for people who want to study the lactate assays for either like a suspension 3D cell culture or like organoids for the complicated structures in organoids.
Yeah, we have a lot of questions, but we'll try to go through them. We got more. What is the stiffness of VitroGel hydrogel matrix and VitroGel stem? Which would fit best for brain, which to bone marrow? For you, John.
So yes, the stiffness on these hydrogel systems is a range. We try to monitor the stiffness based on the ELAX modulus testing. So we kind of measure it with the ELAX modulus. So it covers around from like 50 to like 300 pascals in the ELAX modulus. In terms of stiffness, it's around like 100 to like 900 range. And for different organoids, I have to say is that they are different. Their performance is different. We do have people that using for organoids. I think it's number 2 and number 4 that for some of these brain organoids. But we highly recommend that people to use the screened kit, the discovery kit, to test their own organoids. So even it's the same type of the organoids, if the cell source is different, the tissue source is different, it might cause it growth differently. So we really highly recommend that you use the discovery kit to screen it, and also that your cocktails, the medium you use might also cause some difference.
Okay. For you, John, again, I think another question is, I'm considering using hydrogel to revive cells dissociated from organoids in frozen stock. How does the recovery rate with your hydrogel method compared to the conventional method?
So the recovery rates using like these VitroGel organoid recovery solution to taking the organoid out from our hydrogel is pretty high. I would say that because that we don't use that medium for long terms, it's only take like 2 to 5 minutes to dissociate the whole matrix. So you can get the very, very high yields of the organoid out of it.
Okay. Is there any optional for growth factors free cell culture so that we can add growth factors with media? Also, what is the stiffness of your matrix VitroGel without cells?
You mean the growth factor free medium?
Yeah.
Yeah, I think so for all our hydrogel systems. So what we provide is the pure hydrogels, the matrix system. And what customer can do is that they can use their own cocktail or like use or their own like a basal medium. And with that basal medium, they can add whatever growth factor they want. So the idea is that with the well-defined gel and well-defined the cocktails in the medium, you can create a well-defined 3D microenvironment for the cell to grow, and you can get the full control of this system. And what's the second question? Sorry.
What is the stiffness of matrix VitroGel without cells?
So yeah, again, the hydrogel system. So we create. We have the hydrogel we have here is around like typical range from like 50 to 3,500 Pascal range. That is what we have in these ready-to-use hydrogel systems that as we present here is all in the blue cap is what we have as the ready-to-use hydrogel system. But we also have another hydrogel system, what we call the VitroGel high concentration hydrogel. And with that high concentration hydrogel, actually what we put is the hydrogel with the higher concentration, and it comes with a dilution solution. So people can adjust the concentration of the hydrogel by mixing these two to get a different ratio of the dilution and get a different mechanical strength range. So with that, the typical ELAX modulus on this high concentration hydrogel, you can get it from like lower than 10 Pascal to all the way to 4,000 Pascal. That is the cover a pretty good range for different cell types. And we can also customize the hydrogel to get it over 20,000 pascals on these electron modules, which is close to 60,000 pascals in the stiffness. If people need the even higher systems, we also have our bio-ink system, which will create the gel with some very high mechanical strength to holding the 3D structure with just simple extrusion methods to print this hydrogel. So yeah, as mentioned here, is that the goal is to have the full control. So in terms of the mechanical strength, we are able to using the hydrogel and create a very wide range.
Jenny, this might be for you. How is the growth of iPSC and Hydrogel ECM compared to Matrigel and Cultrex?
Okay, so a couple things. So I mentioned the morphology changes. So it's a little bit difficult to compare side by side because we're gonna have a different morphology. So you're not gonna see like a surface area growing in the same way because it might be growing actually also as a 3D clone. So that's the first thing that I wanna point out. But the second, and to answer that question, once we got the right ratio between the VitroGel solution and the media with the growth factor, they both growing of the colonies was quite similar. Again, taking into account that one method, the cells are growing on a flat surface, there is no third dimension there, not even like 2.5D dimension. While the other one is more of a 3D structure. So if we just look on a bright field microscope, we might be missing part of the growing on the that Z-axis.
Okay. Another question, maybe for anyone, but is it possible to measure the volume of the organoids since it is 3D?
That's a good question. What we typically do is in-house is that we have a microscope system, and those microscope system has the 3D imaging function. So we do a Z-stack, and then we can do like 3D reconstructions of these whole imaging. And with that, you probably can calculate the volume of the organoids. Yeah, that's what we typically do in-house.
Yeah, so I would say that any 3D culture, one of the challenges like characterizing like the full size in 3D. And yeah, it ultimately goes to the microscope that you're using and the technology that you're using, like as John mentioned, like I assume that if you're using a confocal microscope that you have like, you can go deeper on the Z-axis, you can take different planes of the pictures of the organoid and put them together and reconstruct it as a 3D structure. There is actually also newer technology. I recently was looking into is like tomography, and it also let you look more into this like 3D characteristic of your culture. So yeah, it is possible, of course, with some limitations, but yeah, you just have to look into what is the size of the organoids that you're trying to characterize, if it's too big, and what is the limitations of the microscopy technique that you use.
I think we'll have two more questions, and then all the questions that are being asked here, we do have your information. We could come back and answer them individually because we do have a lot of questions. But I'm gonna do two more here. Is the matrix stable in long-term culture, John?
Yes. So the matrix can last for quite a long time. We depends on the cell types, of course. I mean, it's like some cells they will produce some enzymes, and that might eventually slowly degrade the hydrogel system. But in terms of like compare with some animal-based ECM, the synthetic hydrogel system is much more stable. We can keep the organoids or cells growth in this hydrogel for long term.
Okay. There's something that I want to highlight about that. Like, for instance, today I showed the dump method, but we could also just we don't need to restrict it to this dome. We can do it like in a whole well, and like we use the whole surface area of the well and just have the hydrogel covering all that. And you have more space for organoids to grow. And because it's stable, in our experience so far, we can leave the cultures for a little bit longer. But yeah, it also depends on the cells that you start with.
I'm going to end this Q&A with two more related questions about staining. So, John, I think this is, we can answer this one. Has a hydrogel already been tested in immunostaining? And then the other question probably coincide with that is, let's see here. Do you have any protocol how to process the Matrigel embedded organoid to do IHS sectioning for HE staining?
Yes, for both, we do have a standard protocol. And one good thing is that for the immunofluorescence staining, you don't even need to take the cell out of a hydrogel. And all these antibodies and these dyes that penetrate into the hydrogel very well. So you can use the standards, our standard methods to fix the cells and gel together and add all these antibody. We do have some tips on our standard protocol to get the higher these binding rate and get the better imaging. But in terms like, do we have the standard methods? Yes. And can you use this hydrogel with the cells inside for both the histology studies and then the immunofluorescence staining study? Yes. And the imagings actually you can see on our website for most of these imagings that we're taking the cells imagings directly inside of hydrogel. So you don't have to take the cell out, and it performs very well.
Thank you, John. I think there's one last question. I think there's one related to they wanted to, extract, analyze a protein that is organoid culture in a protein. Normally, the culture method is some different way, but how is it different in organoid or analysis of proteins that are secreted by the organoid with the hydrogel system?
That's very good questions. And actually, we currently have the users that really like our systems. The reason is for the downstreams, because that for these the proteins, the assay, it's quite difficult to get rid of these background noise from the animal-based ECM because there's so many protein inside. So with this pure synthetic system, the downstream become much easier. And two things I want to mention is that if you want to just lysis the cell to get like protein out or get the DNA and RNA out of the system, you don't even need to like separate the gel and the cells. You can add in the lysis buffer and lysis everything together to get these out. So you'll get very high yield. And second is that the background is quite clean. So that is become a great benefit when people try to do this for the downstream analysis with the protein assays or other different assays. By saying that, the hydrogel system is not just support like the cell growth or the stem cell or organoids from like stem cell. It also supports these like a tissue fragment. So people can embed in the tissue directly inside of the hydrogel and do the co-culture system. So that is another thing for the downstream is that using these hydrogel system to embed in like patient-derived xenograft organoids or these like tissue fragments, the organoids directly from the patient and embedding in this hydrogel. And then you can do like immunocell co-cultures or like a drug screening directly with this hydrogel system. And also it's compatible for all the different downstream analysis methods.
Well, that concludes our webinar sessions. We do thank everyone for attending and all the great questions. For those that we could not answer, we will get back to you. We do have your contact. Really, again, thank you, Dr. Yanni Piero. Yanes, great presentation, and we look forward for future presentations as well. Please follow us on LinkedIn. And any questions, do have the contact to Dr. Yanes, and we will also send a recorded webinar for you to watch in the end. Thank you so much, everyone. Thank you.